So next topic in this is LOH, that is loss of heterozygosity. What it is? It is again one of the genetic problem which it is there. Normally in given diploid cell, that is where one haploid unit from mother and one haploid unit from father has been there. So there are two copies of genome, one from each parent, that is, and that makes a pair of a chromosome. Each human copy contains approximately 3 billion bases, that is, it can be A, it can be guanine, it can be cytosine, it can be thymine, and these bases are there, they are arranged in a sequence, as we have seen, we call it as a genome. At certain places there can be slight change in a genetic coding. So that is one example, instead of thymine the other pyrimidine is there, or instead of adenine guanine is there. So like that one or other base is changed. So that is going to change the codon, and it may appear only in one parent. It may be in mother or it may be in father's genetic structure. So this is called as SNP, that is single nucleotide polymorphism. This SNP can be because of any reason, because of either genetic makeup of that particular parent, or it may be happening because of whatsoever changes which are occurring during the meiotic phase or during the subsequent replication situations. But this SNP is there, that is single nucleotide polymorphism is there. Now what it is there, that suppose that because of that there is a change in the nucleotide system, and that is going to change in the final output, that is protein. So that is that will have two varieties, one from parent, one from one parent, one from other parent. So when these genome copies which are derived from each parent have different bases, and because of this polymorphic region or SNPs, this region is called as a heterozygous, because one is having one set of nuclear coding, and not in all cases, at one or other places only. At one or other place of this gene it is there, and most of the chromosome within these somatic cells of that individual which are paired. So what happens because of this pairing, if one is not proper one, or because of this polymorphic nature, if it is a different, these SNP locations are heterozygous particularly, and because of heterozygosity the correct protein is formed, or it is also formed, means both are formed. So it does not make much problem. But when there is a loss of heterozygosity, what happens is the total copy of either any parent, either mother or parent, there is a loss. So if it is loss, earlier it was heterozygous. Why it was heterozygous? Because there were two copies, means there were two different sequences. The mother copy of genetic this one material was having some different sequence, and the father was having some different sequence. And since there was a difference in the sequence for the common protein, the protein was common, but still there is a sequence is different. So we call it as a heterozygous. So heterozygous will occur only when there are two copies. But when one copy is not there, one copy has lost, so that is called as a loss of heterozygosity, or LOH. And it has been found in certain diseases. In cancer particularly, it has been found that heterozygous state is required in some of the cancers, and absence of functional tumor suppression gene copy is there, and since tumor suppressor gene is not there, because of the loss of heterozygosity, these people are more prone for the cancer. Apart from that, these are also found in retinoblastoma, another disease where this is there, breast cancer, and in certain conditions this particular LOH has been found. So this is what LOH is, and this LOH is responsible for certain diseases, as we have seen, cancer and retinoblastoma, etc. So the next one is what are various what are other various situations into it. So let us see about it, that is, what are various abnormalities associated with the chromosomes. So again, we have seen some of these last time, today we will see few. So these chromosome abnormalities are there. We have seen earlier that what are there. These are because of various reasons. It may be because of three major single chromosomes are there, in that mutations takes place. It may be deletion, it may be duplication, it may be inversion, and these mutations are take place into this. Already we have seen examples of that some part is inserted in this gene. We call it as an insertion. Okay, then that this portion of this is inserted here, so this has become like this. Two major chromosomal mutation, and other is translocation, that is alteration. This comes to this and this comes to this. So this kind of thing is called as a translocation, that we have seen earlier. There was a figure. Then we have studied about aneuploidy. That means there are abnormal number of chromosomes. That means instead of 46 chromosome, it can be 47 or 45 chromosomes. So these are various seen. Again, in that aneuploidy we have seen trisomy, tetrasomy, etc., and we have seen that they are responsible for certain diseases. Example, trisomy in human is responsible for Down syndrome, which is a developmental disorder which is caused by extra copy of chromosome. I even gave you the example that one of my student he told me about this detection. So these are there. Then what are other structural and abnormalities are there? So this can be deletion, that portion of the chromosome is missing or it is deleted, that is called as a deletion. One of this is example of this is known as Wolff-Hirschhorn syndrome, which is caused by partial deletion of the short arm of the chromosome number 4, and other is known as a Jacobsen syndrome. It is also because of the terminal 11q portion is deleted. We have seen that these PQ portions, chromosome number 11q, that means one is P and another is Q. Then there is a duplication. Some of the portion of the chromosome is duplicated. It results into extra genetic material. Example of these disorders are like Charcot-Marie Tooth disease, which is type of 1, which may be caused by duplication of the gene. That includes that causes encoding of the peripheral myelin protein, which is because of the chromosome 17. I think if you remember, we have seen all these chromosomes number and at least one disease of each. Then we have seen the list of whole big list of genetic disorder that so far whatsoever genetic disorders are known. So these are all examples which are appearing into this list. Then next is translocations are there. There are two things are there. Example is a reciprocal translocation, that is segment from two different chromosomes have been exchanged. Robertsonian is another translocation, that is an entire chromosome has attached to other another adjacent tremor. And in human this only occur with chromosome 13, 14, 21 and 22. So this is one more. Inversion is another problem which can occur, that is portion of the chromosome that has broken up, or it is turned upside down, or it is and then reattached, but while at reattachment the genetic material is invert, means it is broken, and the portion which was towards telomere that comes towards centromere. So that is turned upside down is there, and because of that whole genetic material, the sequence is totally reverse, it is inverted. Then insertions, a portion of one chromosome has been deleted from normal place and inserted to the other one. The difference in insertion and translocation is what? In translocation there is a mutual, that is portion from one comes to other and others goes to this one, but in insertion only it goes from one to other. There is no reciprocal, or there is no coming back, okay, no exchange actually. It is insertion is one side while translocation is both sides, means there are two chromosomes are making it. Then ring formation we have seen, an isochromosome we have seen. So these are various situations are there because of which makes changes in chromosomes, and they are responsible for various diseases. as we have seen that we don't have much treatment or any treatment for such kind of thing. Again, the studies are going on what results into that. There are some environmental or some other chemical factors are involved. Definitely genetic factors are there which are involved, which are responsible for that, and therefore these are there. This happens because of DNA damage during spermatogenesis, or it is because of certain drugs or chemicals which are making it. Then this example of this is melphalan is a alkylating agent which is used in chemotherapy, and that can cause changes. It can make the changes in the genetic makeup, and there are many such drugs which can do it. Will not go for this detection and all, how it is detected, but there are many. Their example we have seen fish and other technologies just recently in last class. So these are various technologies are there by which we can take samples from various places, like amniocentesis means from the amniotic fluid we take, then chorionic virus sampling from fetus, and at many places sample is taken and various tests are performed which gives idea about what kind of genetic abnormalities it can have. So this is another thing we have seen, that what are various chromosomal problems. Then next is we shall see that what happens in the genetic changes which are brought because of one or other reason. So these we shall be seeing, these we call them as a mutation. Already you might have studied it in microbiology, biochemistry, what mutation is. So it is an alteration in the nucleotide sequence of a genome. The genome whatsoever sequence was there, or parents sequence was whatsoever is there, that will be now changing particularly, and which may continue in the next generation. So this is because of many reasons. It can be because of the errors during DNA or whatsoever viral replication. It can be during mitosis process. It can be during meiosis process, and because of other type of DNA like pyrimidine dimers are because of the exposure to ultraviolet radiations, which may undergo error prone repair. So repair takes place, but as if you remember, we have discussed it earlier that just like in tissue repair, we get fibrous tissue formation. So although there is a repair, but it is not having a functional. Similarly, here also because of certain damage, the repair takes place, but it is not a proper one, and then it can be a problem. So these are various reasons why these are occurring. Now we can have variety of mutations. Very important thing is what are the repercussions of mutations. So first thing is it may or it may not have any discernible or observable. Discernible means observable changes in the characteristics, that is phenotype. We know that whatsoever genetic structure is there, and on that basis there is observable results are there. We call them as a phenotype. Okay, so this genotype, the changes in the genotype may or may not produce any big changes in the phenotype. It may play part in both normal and abnormal biological processes during evolution, cancer, and development of immune system, junctional diversity. Mutations are basically ultimate source of all genetic variations. So any genetic variation which are occurring from first generation, from one generation to other generation, these are basically because of the mutations. Mutations can result in many different types, that changes in the sequence of the codons or the genes. It can, as I told you, it can have no effect. It can alter the product of gene, and that product may be more effective or more beneficial, or it may be less effective, less beneficial. So there is a wide range, no effect, better than previous, and worse than previous. So in this sequence, anything can happen because of the mutations. So if the mutation changes a protein produced by a gene, the results can be likely to be harmful, with an estimated 70% of the amino acid polymorphism that have damaging effects. if others may be neutral or marginally beneficial. So because of this effect, the mutation have on the genes organism, the DNA repair is important, but it may not be possible all the times. So there are variety of mutations are there. They involve in the duplication of the large section of DNA through genetic recombination. Now this technology is used, we call it as a genetic recombinant technology. We shall be seeing about something about this recombination and recombinant technology. So it is a duplication of large section of DNA. This DNA are specifically put into certain microorganisms so that they can do that job. So here these mutations which are, these are the deliberate mutations are there. So these are happening there, and they are responsible for certain protein production which are responsible. Now we shall see what happens. We have already seen chromosome number can be there, and there can be example, we have seen aneuploidy, trisomy, tetrasomy, etc. So this chromosome number can be changing, and while changing it, it can also have mutations simultaneously. So these are various things which are there. We shall see various mutations. Mutations can be lethal, it can be non-lethal. It can be it can be problematic, it can be non-problematic. So variety of these mutations we shall be seeing why these are there. So basically, why mutation takes place? There, it can be mutations can be happening spontaneously because of the molecular decay, or they are due to error-prone replication bypass of the naturally occurring DNA damage. So whatsoever DNA damage is occurring, we have discussed it earlier that we have got very nice DNA repair system, and without DNA repair, when everything, we have got so many checkpoints, if you remember, during cell cycle. So these checkpoints, they ensure that there is no damage. However, somewhere if there is a failure of these checkpoints, all these proteins, if you remember CDC and CDKs and various proteins, we have seen it during our previous section of cell and cell development. So here these are important, that if this is failed, then this error-prone replications that bypasses the repair, and this is resulting into a damaged one. Then there are error introduced during DNA repair, as I told you, just like we have got a fibrous tissue during tissue repair. Then induced mutation, they are caused by certain mutagens. There are many chemicals or environmental factors, they are known as mutagens, and these mutagens are responsible for having changes in the DNA, therefore we call them as a mutagen. Many drugs, many chemicals are known as mutagens. So these are then at some time scientists also deliberately introduce mutant sequences through DNA manipulation for experiments. If you know, or if you have gone through various articles during this COVID-19, people say that this virus is a man-made virus. People say that the scientists have inserted some code, some genetic material in such a way that we cannot have treatment for that, and this is how these scientists are doing. There are many, and there, if you remember, we have discussed about a few words like knockout or genetically modified animals, which are used in the research. So these are the mutations which scientists are doing deliberately. And these terms, if you remember, genetically knockout, that particular some gene which is there, which is controlling for or regulation of fat. If it is removed, then these animals will become fat, because this gene, which was responsible for control of fat or diet or control of what we can say hunger center, that will be, or satiety center rather, that will not be there, and this animal will eat a lot and then become fat always. So these are various ways why there are mutations are there. So let us see what are the impacts of this and how these mutations takes place. One of the study says that around 66% of the cancers causing mutations are random. They are randomly occurring. 29 are because of the environmental factors. maybe plastic or the chemicals which are being sprayed on food material, and presence of this fertilizer etc. in food. So these are various chemicals or environmental toxicants, particularly industrial waste which is there either in water or in air, and 5% are inherited. So this 66 are random, means they were not there in previous generation. There is no impact of environment, still they are causing, and no one knows why it has caused to them. Humans on average pass 16 new mutations to their children, but fathers pass more mutations depending on their age every year, adding 2 new mutations to a child. And this is one more thing, that if this the childbearing age is the best is up to 30. If something childbearing is goes beyond 30, then the problems of genetic problem, people say that they are more, or scientists say like that. So what are various mutations? Spontaneous means they are occurring spontaneously, all of a sudden they are occurring. They occur without non-zero probability, even given healthy uncontaminated cell. I told you these are random, no one knows, all of a sudden it occurs. Naturally occurring oxidative damage is estimated to occur in 1000 times per cell per day in human, and around this 10,000 times, 100,000 times, that is 1 lakh times per cell per day in rats. So spontaneous mutation can be characterized by following type of specific changes. One is tautomerism. If you studied biochemistry, you must be knowing that because of the structural specificity, there are only specific bindings are possible. These are because of A to G, that is adenine can bind with the guanosine, and CG, ATCG, we have seen adenine to thymine, and cytosine to guanine. So this ATGC pairing is there because of the specific bonding which is possible, which is there. Now what will happen? These are because of the hydrogen bondings which are there. Now if there is a hydrogen atom is reposition, if it changes the position, then the hydrogen bonding will change. Because of this hydrogen bonding, it is one of the force that will change, that will cause mispairing. That is, instead of normal ATGC or AT and GC, it can be something like that. Instead of AT, it can be AG, okay, or it can be AC. It will not be AT. Instead of that, it can be AC or AG, and vice versa. So like that, it is an incorrect base pairing that occurs during replication. This is called as a tautomerism. So because of this, now this hydrogen bonding can change because of, again, there is an internal environment, or what we call as a homeostasis. So because of any homeostatic changes or presence of any such chemical nearby cell, this can happen. Then another type of reason can be depurination. There is a loss of purine base, that is adenine or guanine, to form apurinic sites. So these apurinic sites are there. Then other is deamination, that is hydrolysis, changes the normal base to atypical base, which contains the keto group in place of the normal amino group, and therefore it will change the structure. The cytosine will behave like this uracil, and the adenine will behave like hypoxanthine, and which can be corrected by DNA repair mechanism. But if it is not corrected, and another is 5-methyl cytosine, which will behave like thymine. So these are less likely to be detected as mutation because thymine is normal DNA base. This is actually this type of specific, or this type of things are we are using in antiviral or anti-cancer treatments, when we will see that pyridine pyrimidine analogs. So we have got similar analog. They will getting incorporated during cell division, and they will fall. They will make the false false product. Then next one is slip strand mispairing, the denaturation of the new strand from the template during replication, which is followed by renaturation in different spot, and this can lead to insertion or deletions. So these are various spontaneous mutation. Then other mutations are there that are error prone replication bypasses. So So here there are translation synthesis which are taking place in certain organisms. Then there are errors which are induced during DNA repair. I told you very nice mechanism is there, but somehow it is not working properly, and this will cause erroneous DNA and mutated one. Then other are induced mutation. These are because it come with the contact with some mutagen or environmental factors, and these are example of this. These are various chemicals are there like hydroxylamine, then bromodioxyuridine, alkylating agents which are part of cancer treatment, which are very important in the cancer treatment. So these will cause mutation, and therefore they will be used in the treatment of cancer treatment. Then various agents which are forming DNA adduct like ochratoxin is there. Then agents which are known as a DNA intercalating agent. Ethylene bromide is used for staining of DNA, and even alkylating agents can also do the intercalation of the DNAs. Then there are DNA cross linkers which will find the cross linking or covalent bonding, so it will not allow the DNAs to separate it. It will form a very strong oxidative damage. The oxidative radicals will be causing damage. If you have studied oxidative damage earlier somewhere in biochemistry, or example, very nice example of it is rancidity of oil, and in that rancidity of oil somewhere maybe 11, 12 somewhere you might have studied that one molecule gives rise to so many molecules of free radicals, and these so many molecules, it is exponentially growth, 1 has to 2, 2 to 4, 4 to 8, 8 to 16 like that. So this is known as exponential, and this causes very high damage because they are very reactive and can change the chemical nature of the compound. Then other is nitric oxide. It is also one of the free radical, and it can cause problem. Then radiations like ultraviolet radiations, ionizing radiation, they can affect and they will cause oxidative damage and changes in the pairs. Then one more way of classification we have seen, one is spontaneous, that is the classification was based that how it is taking place. So whether it is taking place because its own, that is spontaneous, or because of some inducer, that is which are induced by a certain mutation. Then another type of classification we can have, that is what are the effect on the structure. So what we have studied all these insertion, deletion, translocation, etc. These are also mutations. So these are various mutations which are taking place. Then there are another small scale mutations which are there. These are called as a point mutations because it is occurring only at one or other place. And example of these are very commonly they are known as a indel mutation. IN in DEL del. Indel means insertion and del means deletion. Okay, so this is only and changes are there only because of one or other nucleotide. But you can imagine if one nucleotide is changed, it can change whole sequence of the DNA. They are usually caused by transposable elements or the errors during replication of repeating elements. Insertion in the coding region of the gene may alter during splicing of the RNA. So some of these are known as a splice variants. Okay, splice variant means while splicing, if you know intron exons. So while this removal of introns, this if this cutting is there, splicing takes place at slightly one or other here or there, one or other nucleotide here or there, then that will cause change in the whole sequence. So this will cause shift in the reading frame, and therefore it is called as a frame shift mutation. You can imagine there was a sequence, suppose A, T, G, then G, whatsoever you make it. So there were three like that it was there. Now what has happened that the RNA is formed. Now after ATG, the next was intron, it has to be removed, but instead of either if it is getting removed before G, so only AT is remaining, or after G, so ATJ, next is U, next is suppose C. So instead of that it becomes ATGC, it should have been only ATG. So it becomes either AT or it becomes ATGC. So one nucleotide is changing, but because of that we know for each 3 there is a codon. It is giving some information. It may be either start codon, it can be stop codon, it can be either for any kind of amino acid codon. So this is causing change in the reading frame, and that is therefore called as a frame shift mutation. These are this causes the significant alteration in the gene product. Deletions, just like that, deletion can remove 1 or more nucleotide, but it is less than 3, okay, or instead of 3 it can be 2, 1 or 4, 5, like that. So it will cause change in the amino acid, and if it is making it, this deletion will also change the sequence, okay, if only 1 or 2. Then substitution mutation, it is caused by chemicals or malfunction of DNA replication. Exchange of single nucleotide for another, it will take place. These changes are classified as a transition or transversions. Example is the purine is exchanged for pyrimidine. Purine is exchanged for purine, that is instead of A, G is inserted, or for pyrimidine, for cytosine, thymidine is inserted. Okay, it should have been C, but now it becomes T. So it can be caused by nitrous oxide. It can be caused because of other chemicals, and it causes base mispairing or mutagenic base analogs, like we have seen BRDU, that is this uridine derivative, then bromo D uridine derivative, and less common is a transversion which exchanges purine with the pyrimidine. There are 2 we have seen. One is a transversion and a transition. Transition is having exchange of purine to purine and pyrimidine to pyrimidine, while transversion is purine to pyrimidine and pyrimidine to purine. So adenine will be having conversion to cytosine and like that. So these are the various mutations. These are because of substitution, that is adenine will be substituted for guanosine, or cytosine will be substituted for thymidine, anything like that. So these are called as a substitution mutation. So you can imagine, it is very easy to understand that every 3 codons are important, and if that sequence changes, then it can become anything. It can become starting codon, it can, even though there is a no need, still it can be making a start codon, it can be making a stop codon, which will be causing stoppage of this gene expression. It can code for a different kind of amino acid sequence than the normal one. So these are, it can change the spaces or sequence of introns and exons, and whole structure can be there. So it can occur in the coding region of the genome. They are more likely alter their protein products, and it can be categorized by their effect on the amino acid sequence. Frame shift mutations, they are caused by insertion deletion. We have seen this, and these are the code example are there. Example, just a minute. Hello. हाँ, लेक्चर सुरू आहे, 10-15 मिनिट. हां, चालते आलं लेक्चर जाऊन. ओके, लेक्चर संपवतो. हां, so it can be anything, and because of that the example is given nicely. If there is a change, if the sequence changes, that is CCU, if the U in the CCU is deleted, here CCU is there, so it will become CCG. Now this is CCU become CG, then it becomes ACU. So whole sequence has changed, that has become a frame shift mutation. The frame which should be CCU, GAC, UAC, that has changed to CCG, ACU, AAC, ACC, UAX, and that will be instead code for the amino acid sequence will change now. It will code for instead of proline, aspartic acid, tyrosine and leucine, it will code for other amino acids. And sometime it can also make a stop codon. Because of that, the protein will be much different structure, because it will be very shorter structure if in between stop codon comes. So this is what the impact of the frame shift mutation. These mutations therefore making a problem, and they can change whole sequence, and therefore a new protein with different structural activities are there. So there are this point substitution result in the change in the signal nucleotide, and it can be either synonymous or non-synonymous. Now what is synonymous? It replaces the codon with another codon that codes for same amino acids. So it is that way. That means that has not made a big difference. Example, if you see just above example, what above example says, it was CCU, GSU was there. CCU was making for what? Proline, but it has become to CCG. Again it is coding for proline. So CCU and CCG, they are having synonymous substitution. That means proline to proline. If this occurs, then there will be, even though there is a mutation, it will not make any change. I hope you are understanding this. Okay. So this will not make any change. And non-synonymous, this synonymous mutation occurred due to the degenerate nature of the genetic code. But if this mutation does not result in any phenotypic effects, then it is called as a silent mutation. That is no impact on the phenotypic. The body function will remain same. Okay. But not all synonymous substitution are silent. There can be some silent mutation in nucleotide outside of the coding regions, like introns, etc. And therefore, although it looks synonymous over there, because of the impact on the introns, the overall it will change the formation of messenger RNA, and therefore the changes can be there. The other one are the non-synonymous. The replacement of the codon with another codon that will code for a different amino acid, and because of that the produced amino acid sequence is modified. This non-synonymous substitution can be classified as a nonsense or missense mutations. Nonsense means in the change that changes the nucleotide, that causes substitution of a different amino acid, and it can make the resulting protein non-functional or change in the function. Rather, we'll use better word is, it can make the protein with a different function. As I told you, in certain cases it may increase the function, and these mutations are responsible for diseases. There are many diseases are there, like sickle cell disease, etc., etc. If the missense mutation occurs in the amino acid codon that result use of a different but chemically similar amino acid, then sometimes little or no change, or rather even change in increase in activity can also be there. So this is the impact of the missense mutation. Missense means the new protein is formed, the change in the structure of protein, while the nonsense mutation means what will happen is, because of that mutation it will result in the premature stop codon formation. So in nonsense codon will cause the premature stopping of the genetic expression, and this nonsense codon in the transcribed RNA, that will possibly truncated, and that will form a non-functional protein, that will be very small protein than the normal one. The sort of mutation has been linked to many diseases like congenital adrenal hyperplasia, etc. There are many such diseases are there. Now there are many functional problems it may cause. So this function, by function wise, if we say that loss of function, that means because of these changes, as we have seen above, there may be loss of function. So one or other function is not there. So there are many diseases are there. Example is Muller-Morph disease, etc. Then another one is, it can gain of function mutation. In certain cases there can be increase in the activities. I told you, even there is a replacement of amino acid which may make the protein more active. Then next is dominant negative mutations. They are also called as antimorphic mutations. They have an altered gene product that acts antagonistically to the wild type of allele. Now one important thing is what are known by wild type of allele. Wild type means when first time it was found, whenever the researcher has studied it, then they will find it that this type of genetic structure is there which is giving rise to this kind of protein. So this is known as a wild type allele. Then later if we study anything, then if we find any different, then we will call it as a mutation, okay, or variant or polymorphic form, whatsoever we can call it as. So it can be polymorphic form or it can be, so wild type is, it may be lesser in number, but it was found first time. Polymorphic may be more in number or it may be rather more active, but then it has been formed, it has been observed later, so we will call it as a polymorphic form. So wild type means it was known first time, which was found first time, particularly it was recognized first time for given protein or given amino acids, like that. So dominant negative mutations are there where the altered genes product that acts antagonistically to the wild type of allele, and this mutation usually result in the altered molecular function. Mostly it causes inactive, and they are characterized by dominant or semi-dominant phenotypes. So there are many cancers are there, particular changes in the gene P53 and many such PPR gamma, etc. Marfan syndrome is also caused by mutation in various genes into that. Then other are hypomorphs. So these are characterized by altered gene products that acts with the decreased gene expression compared to the wild type, and usually hypomorphic mutations are recessive, but it can still make changes, it can still affect it. Then neomorphs are characterized by the control of new protein product synthesis. Lethal mutations which lead to death of the organism which carry the mutation. These are again mechanisms nowadays being made for killing certain species, that mutations are done into that so that it will cause destruction of that species. So this was a classification on the basis of the functions, what was, how it changes the function. Basically most important thing is there, whatsoever we have done, it can cause total loss of function, it can cause gain of the function. These are two important, or there is no big change in the function. These are three things can occur because of that. And same is the thing, fitness, it can be harmful or deleterious effect on the fitness of the organization. We have seen so many diseases for that. It can cause a beneficial or advantageous mutation which increases the fitness. Example, this is an very important example why it is beneficial, because this kind of mutation has a lead for adoption, isn't it? We have got for adaptation when we say, or we say evolution, that from there are lot many changes, environmental changes. Therefore suppose those like us who are living in the warm atmosphere, we have got black skin, we have got dark colors, we can withstand with the hot summers with 50 degrees Celsius, while the people who are living in maybe Siberia, where minus or negative temperature goes, they are adapting themselves. So this kind of adapting behaviors are there, they can occur because of the their same thing we shall study how beneficial or important mutations are there, particularly for microorganisms which are forming resistance. So this is beneficial for that organism, because this organism develop mechanism and it passes it to the next generation, and thus this become resistant to given antimicrobial agents. And it can be neutral, it has no harmful or no beneficial effects are there, okay. These are occurring and they are not even noticed also, but they are occurring. So these are all various kind of mutations are there. So we have seen all the mutations, how these mutations are there, and accordingly what are their impacts. So this is the topic where we have finished all about various mutations, what are their roles, why they are causing, what do we mean by polymorphism, and there are multiple ways by which there can be changes in the genetic structure, either at DNA level or it can be at RNA level, and because of that the three impacts we have seen, it can be either decrease in the activity or stoppage of the activity, it can be lethal, it can be improvement and which is useful in fitness, increase in the fitness, or we call as evolution or adoption, and there may be no big change which is seen. but because of this polymorphism or because of this mutation, the next generation may have impact on that if other chromosome is having some again different structure, and then we can see that. So maybe the earlier generation did not have any big observable change, but because of that faulty gene it can have in the next generation. So this is how inherited disorders are there. There are many inherited disorders are there. Many diseases are there. They are present in carcinogenesis. I have seen, we have seen about various resistances, etc. So this is what mutation is there, and how the mutations are important, that we have finished. Okay. So once I hope, I think you should take some time for going through this material, because lot many lectures we have finished. If you have any discussion or difficulties on that, you please go through this. Already site is known to you. This is I feel based site, which gives very important and precise information. So there is you can go through this, and if you have any difficulty you can ask anytime. So take care, be safe.